It's a process which gives rise to an altered cell type. So the easiest way of understanding where it's important is to actually look at the cells which make us. So we're made up of skins. There's cells that give rise to our different organ types. We have liver cells, we have neuronal cells, we have skin cells, etc. But when we're first formed at birth, we form from one single cell, which is the formed from the gametes from our mother and our father. That single cell has to give rise to all of our different cell types in the body. But all those cell types have the same DNA, because they've all derived from the same source. So why do we get different cell types that make up us as individuals? And those different cell types arise from genes being turned on and off in different cell types, and those genes are turned on and off as a result of epigenetic processes. So they're vitally important in determining our formation right from our first moment of birth to our adult phenotype, and then changing throughout life as well as we adapt to the environmental influences around us. We don't know exactly how environmental influences really perturb that epigenetic code. And we have to be careful to distinguish the epigenetic code from the genetic code, because the genetic code could be disturbed by environmental mutagens about which we know a great deal. We're talking about the non-genetic code, so the epigenetic code. But we do know from some studies, for example, in what's called the twin studies, where you've got monozygotic twins that at birth and throughout their lives have exactly the same genotype, but over time, as they age, they develop a different phenotypes, different physical characteristics, and those differing physical characteristics are not due entirely or not due to their genotype, because they've got the same genotype having developed from the same egg, and they're thought then to be due to different epigenetic changes that have been modified by their environmental conditions over time. Now, quite how those changes are related to the environmental conditions, how they give rise to the altered phenotypes, is not something we fully understand at a basic biological level at the moment. There's a great deal more to be done there in order to fully understand what's happening. Epigenetic effects could play a role in adaptation, for example. And we don't know this definitively, but it's a good hypothesis. As we alter and adapt to a changing environment, that we do that by changing our epigenetic influence. And this is the whole basis of a foundation of the genetics called Lamarckism, that we adapt to an environment, and that adaptation is passed on to our progeny through the epigenetic change. So this is different to Darwinian genetics, where you're talking about the environment selecting for the fittest, so people who've acquired a phenotype as a result of a changed genotype, which gives them an advantage in that environment, allows them to survive, which is quite a long process taking many generations. What we're talking about in epigenetic change is an adaptation to an environment which could be advantageous or adverse, that can occur within one generation and then be passed on subsequently. And it is considered possibly to underlie some disease phenotypes, where we've seen some diseases rising in populations that can't be explained by Darwinian genetics, because they're just happening within one generation. It's just too fast. And some of these are thought to have some epigenetic component. Lamarckism as a genetic principle was not immediately accepted. So Darwinian genetics from the origin of species has always been cited in schools as the way we've evolved and the way we're selected and live better in our environment. But that's really because there was no biochemical principles to underlie Lamarckism, so it was simply a hypothesis that we could adapt to our environment. Now that we know that these epigenetic marks exist, and there's no doubt they do exist, and we're learning a great deal more about them through the new molecular techniques we have now, we've suddenly got a means of explaining that particular hypothetical way of doing genetics. So we've got a way of explaining it, we've got a means to do it. What we need to now know is to get the associations between environment and epigenetics. And we don't really have those at the moment. We know epigenetics changes, but we don't know what the association is with the environment. People do some academic studies, they do some high exposure studies, they look to see what could be affected. What we need to do in these kind of meetings is to look at those studies and say, well, what's happening? And then what's the relevance for things that we're looking at either with industry or within public health now? Do they have relevance for exposures that you and I might have? Do they have relevance for new products being developed? Do they have relevance for testing methods? Do we need to incorporate them into testing methods? How do we need to look ahead to take account of this new biology and work out what its relationship is to industry and to testing principles and to the way that chemicals are developed, or in terms of public health to what we're exposed to? What's the relationship of these with that, and where does the biology matter? What do we need to look at that's merely of, let's say, academic interest versus that's of practical interest that we may need to take into a sort of more interventionist type approach? So it's very important to bring people from different disciplines together in these fields, the people from the academic side and people from a more applied science side, in order to get that greater understanding as to where the overlaps are and where things need to be taken forward. There's a lot to be understood in terms of the basic biology. There's certainly an awful lot more to be understood at what we might call low-level exposures, so the kind of exposures that we get in day-to-day lives. That's going to require new methods of looking at it, new sensitivities. We need to find better and cheaper ways of doing it, because some of these studies are very expensive. And then from there on in, understanding, I think, potentially some of the mechanisms involved, because that's the only way that we'll start to be able to translate the basic science into a more applied agenda.